Good evening, everybody. My name is Mark Nutter, the Conservation Programs Director here at New Hampshire Audubon. I'd like to first acknowledge that this presentation is streaming to you from our state headquarters in Concord, New Hampshire, which is located within the site of the ancient village of Pennacook in Nadakina, which is the traditional ancestral homeland and waterways of the Abenaki, Pennacook, and Wabanaki peoples, past and present. I'd like to acknowledge and honor with gratitude the land and waterways and our ancestors, the Alnambak, who have stewarded the Nadakina throughout the generations for thousands of years. And I invite you to learn more about the Indigenous presence on the land you occupy by visiting the website native-land.ca. Here you can explore and click on territories of Indigenous people and get connected to resources to learn more. And for an even more in-depth understanding of the Granite State, check out all the educational resources at indigenousnh.com, including this interactive story map that details Indigenous presence and their stories here in New Hampshire. These resources, among others, have helped me recognize the ongoing consequences of colonialism for all people of color and the need for change in our current society. And thank you once again for your interest in tonight's topic, Beecology, a community scientist helping pollinators, by Dr. Robert Gegear, professor of biology at UMass Dartmouth. I'm looking forward to learning a ton alongside you this evening. And as you may know, this talk is the 12th session of a year-long webinar series called Exploring Connections to and Stewardship of the Natural World, supported by a New Hampshire Humanities Council grant. The past recordings of these excellent talks can be found on the New Hampshire Audubon's YouTube page, which are also linked on the series webpage. And throughout this series, we're exploring the intersection of the sciences and the humanities, finding and forging new ways to connect with nature and learn about the importance of conservation action. So I want to invite you to really take the time and space to consider how tonight's topic informs, strengthens, or otherwise support how you define yourself as a person and how you connect with human communities as well as the wild ones. I implore you to reflect on why this topic is important to you and your personal value system, and how you can connect with others through this topic in your daily life. This is the 8th and final talk in a mini series of talks focused on pollinator conservation, and we are delighted to have Robert with us this evening. And in case you didn't know, both of New Hampshire Audubon's year-round nature centers have incredibly manicured landscapes that demonstrate how landowners can benefit pollinators through planting native vegetation. The demonstration gardens at the McLean Center in Concord are managed by Diane DeLuca, senior biologist, and her amazing crew of volunteers. And the gardens at the Massachusetts Center in Auburn are managed and supported by the UNH's Master Gardener Program, and a legion of volunteers managed by both the Master Gardeners and New Hampshire Audubon. And both these outdoor locations are open year-round and can serve as inspiration to you in your landscaping endeavors. And most recently, we're celebrating the unveiling of a virtual tour of our pollinator demonstration gardens, which I will put a link into the chat shortly. And before I hand it over to Robert for tonight's presentation, I'd like to take this opportunity to briefly describe how this webinar fits into the larger mission of New Hampshire Audubon. For those of you who don't know, New Hampshire Audubon is a state-based environmental nonprofit organization completely independent from National Audubon. We rely on members and donors like you to support our charitable mission, which has 4 programmatic pillars: connecting people to nature through environmental education experiences like school programs, nature day camp, and webinars like these; researching and conserving species in peril, including large raptors and small birds; managing about 10,000 acres of wildlife sanctuaries throughout the state for habitat and recreation; and finally, advocating for sound environmental policy in the New Hampshire State Legislature. I am here today because of donors and members like you. We also rely on a huge network of volunteers that assist us with wildlife monitoring, ambassador animal care, environmental education, and wildlife sanctuary management throughout the state. If you're a volunteer, member, or supporter of New Hampshire Audubon, I would like to sincerely thank you. We simply couldn't achieve our charitable mission without you. If you'd like to become a part of our conservation family today, which I hope you will, please check out our website for ways to get involved. We do have over 80 people registered for this evening's talk, so you'll see that we're in full webinar mode. So that said, please feel free to use the chat for any thoughts, comments, and reactions you might have, and reserve the Q&A button for any questions that you would like answered by Robert. For fun, try typing into the chat where you're watching this presentation from. It's been great to see the vast geographies we've been able to reach through these webinars. And so that others may engage with you, simply change where you're sending your chat message to say all panelists and attendees. Who knows, you might just meet your neighbor tonight. Diane DeLuca, the senior biologist responsible for orchestrating this series, and myself will be monitoring the Q&A here as well as on Facebook Live tonight. And Robert will take questions after the presentation. And I've set the parameters of the Q&A here in Zoom so that other attendees can see your question and upvote or even comment on that question that they want to see answered. And that's so we can hone in which questions we have our limited time to answer this evening. So without further ado, I'd like to hand it over to Diane to introduce this evening's presenter.
Thanks, Mark. We are very pleased to have Dr. Robert Gegear sharing his expertise with us tonight. He is a professor in the Department of Biology at UMass Dartmouth and the director of the New England Beecology Project, which is a citizen science-based effort to collect large amounts of ecological data on native pollination networks in New England. He has been studying the neuroecology and conservation of pollinator plant systems for over 20 years and has over 40 publications in peer-reviewed scientific journals, books, and the popular press. In recognition of his ongoing efforts to protect and restore native biodiversity in Massachusetts, Dr. Gegear was awarded the 2018 Regional Impact Award by the New England Wildflower Society. Tonight, he will be sharing a presentation titled, actually, I think you changed the title, so I won't share that out, but he'll update participants on the decline of wild pollinators and the importance of collecting critical ecological information that is needed to develop effective conservation and restoration strategies for threatened pollinator species. The Beecology project was developed to recruit citizen scientists from across the region to digitally collect and submit ecological data on native pollinators. You will learn and practice data collection using the smartphone and web apps developed through this project. So without further ado, we're very pleased to have you, and thanks for being here.
Okay, thank you very much. Pleasure to be here. Thank you for the invitation. Okay, so as Diane mentioned, I changed the title a bit. It's the same theme. I'm going to be talking today about how we are using ecological data to protect and restore native pollination systems that are at risk of local extinction. Ecological data that I'm going to present today is coming from my primary research lab, so myself, grad students, but a good portion of it, or certainly what I'm doing is influenced by the Beecology project that I mentioned, and that's my citizen science project that I developed or started in 2016 or so, because I saw that there was a desperate need for more data on plant pollinator systems, particularly those systems that are being degraded, both from the animal side and the plant side of the equation, which I'll talk a little bit about. When I started, so I gave a talk here for New Hampshire Audubon in 2018. Obviously, back then it was in person. And at the time, we were in the data collection phase of the project, and currently we are in the implementation phase. So we've collected the data, confident that there are very clear patterns that I'll talk about, and we are now creating habitat that, we started putting habitats in across Massachusetts. Of course, I hope to expand this to other states, including New Hampshire. But pretty much immediately, as soon as the plants started to bloom, the at-risk species showed up in good number. And this is the second year for a lot of those sites, and we're seeing numbers at least the same number, or if not more. So it's quite exciting. It's been a tremendous success. You'll notice at the beginning of my talk, with the Beecology project, you think it's all about bees, and actually the Beecology project is not all about bees. Bees certainly play a role, but the focus is more on native plants, animal pollinated plants. And what I hope you'll get from today's talk is that there's a considerable difference between saving bees and saving their functional role as pollinators of native plants. They're completely separate things. And bees are certainly important, but they're not the most important pollinators out there. There are many plants that are targeting animals other than bees for pollination. And the diversity of those animal plant connections, that's really what the Beecology project is trying to do. The diversity is being reduced by as a result of our activities. And so we're trying to figure out what we're doing that's causing these systems to degrade and do things to try to prevent them from becoming extinct, one side or the other, the plant or the animal side, and certainly, stop them from being degraded. So that's what I mean about the more than just the buzz. That the plant is, you're going to see by the end of this talk that it's the native plant, and it makes sense because the plant is being pollinated. We'll start off by talking a little bit about the difference between what a flower visitor is, a pollinator, and a pollination system. If you know what the difference is, you're well ahead of the game, well ahead of most people that I speak with. The term flower visitor and pollinator are used interchangeably, and this is creating a problem, is that we think that we're helping the pollination, but actually we're just helping to save the animal, not its functional role as a pollinator. And so I'm going to spend the first part of this talk going over that difference. And then I'm going to talk about the problem, biodiversity loss, the loss of these pollination systems, and how I've used Beecology and you, citizen scientists, to help define the problem and also to help find solutions to the problem. And then I'll finish with how we're restoring biodiversity, so restoring these connections. I've got hundreds of beecologists at this point across the state and into, moving into Connecticut, Rhode Island, some other places that are putting in the proper plants and that target these at-risk species and helping them. And I'll talk about how that's empowering and what we need to do to assess our own habitats and our own backyards, our own gardens, to see, are they doing what we think they're doing? And if they're not, what do we need to do to fix it? And I can give you suggestions. I have a plant list that's I'll talk about. I have a bunch of handouts I can give. I'll send them to Diane, or you can visit my website and I'll show you the website. So as I mentioned, we're going to start off with the basics. What is a flower visitor versus a pollinator? Wide variety of animals visit flowers to feed on the nectar or the pollen, or both. Most of the animals that do this are insects. We have some examples here. We've got obviously the bees, good number, thousands of bee species, but we also have moths and butterflies. We have flies, wasps, beetles. The vast majority of what we see visiting flowers are insects, but certainly we have some vertebrates as well. Hummingbird shown here. So in terms of flower visitor, if we want to get formal with what we're calling these flower visitors, if they're visiting for nectar, we're going to call them a nectarivore. If they're visiting for pollen, we're going to call them a pollinivore. So as you can see from the background, there are a lot of things in bloom. They could be native, non-native, a lot of floral resources out there for the individuals to choose from, and they need to figure out which ones offer the most nectar, the most pollen. And so the question is, how do they do that? And there's this, especially with insects, the idea that they have small brains and that they're driven by instinct. And if we put out flowers that have a certain color or smell, that's going to attract butterflies in general. Or if we put out another color, it's going to attract bees. That many people think this way, but in reality, as I said, they're looking for food, they're looking for good sources of nectar and or pollen, And so if they find a good source, they're going to continue to use that source. And so we can look to see what role instinct versus learning play in the lab. And I've studied learning and memory in insects quite a while. And so here we have our monarch butterfly that's tethered, touch the legs with a cotton swab soaked in sucrose solution. You get extension of the proboscis or the tongue, similar to Pavlov and Pavlov's dog with the salivation. We show the butterfly stimulus. We can show a color, odor. In this case, it was yellow, a shape. Pair it with the sucrose reward. As soon as the tongue touches the sucrose, it forms an association in its brain. Then during the testing phase, we can show it a variety of colors. So show it a blue. This color is a bit closer to the target color of yellow. And we see no extension of the proboscis and no movement of the antenna. And we show the paired color, yellow, and we see an almost immediate response. That butterfly is looking for that food reward. So, after just a couple of pairings, this butterfly will remember that color for at least 2 weeks. Again, it can learn colors and odors and combinations thereof. So the point of this is that we really need to think about not what the flowers look like, but what the flowers contain. What quality do the flowers? How much nectar and pollen, and we're going to let the animals decide for us which ones have the nectar and pollen that they like, because it isn't necessarily just about the amount of sugar. There are other things in there that the animal might like. So looking at it from an animal foraging perspective, we are going to let the animals tell us what they want, and then we're just going to give it to them, right? So that's the one point that I want to make here. So we've got all of these animals that are visiting, and at this point we're calling them flower visitors because we have no clue what they're doing with respect to the plant. And to change that label from flower visitor to pollinator, we need to know what that animal is doing with the plant. And it has nothing to do with the animal, the term pollinator. It has everything to do with the plant, and specifically plant reproduction. So if the animal happens to transfer pollen from the male reproductive organ to the female reproductive organ, so the surface of the stigma, leading to fertilization and the production of seeds and fruit, then and only then can we call our animal a pollinator. All right. So, plants have evolved a wide variety of different ways to entice animals to visit them, to pick up pollen and deposit it. And this process is called pollination. It's a reproductive process from the plant's perspective. And plants, if the pollen's transferred from flower to flower within the same plant, some plant species can mate with themselves. This is called selfing. Right? Other plants can't receive their own pollen, and they need to receive pollen. So our plant over here, if the pollen's transferred from this flower to another plant, it's called outcrossing. And so many plants are self-incompatible. They can't receive their own pollen, so they need to receive pollen from another individual of the same species. All right, so if we have a self-incompatible plant, even though our animal is transferring pollen within the plant, it is still not a pollinator because it's self-incompatible and it's not leading to fertilization and the production of seeds and fruit. So I'd also like to point out that, flowering plants, the process of pollination can be carried out not just by animals, but there are wind pollinated plants. There are also water pollinated plants. The problem with those two modes of pollen transfer is that they're very inefficient, right? If you want to mate with an individual or send your pollen to an individual that's upwind, then it's not very efficient if you're wind pollinated. Similarly, water upstream, downstream, depending on where your mate is, potential mate, it can be a very inefficient system. So the vast majority of plants are flowering plants, and the vast majority of flowering plants are animal pollinated because the animals, while they're looking for food, will certainly travel upwind, downwind, or wherever they need to go. But as we'll see, things get a little bit complicated. So let's look at an example here. Here we have obedient plant. We have two bumblebee species. We've got Bombus fervidus up here. We've got Bombus affinis down here. Bombus fervidus is entering the flower, making contact with male and female reproductive organs, leading to fertilization, production of seeds and fruit. Our Bombus fervidus is a pollinator. Here we have Bombus affinis. Bombus affinis doesn't go inside of tubular flowers to get the nectar. What it does is it will bite a hole at the base of the flower, and it has a really short tongue. Our Bombus viridis has a long tongue and can stick its head in, and the nectar is located down here. It can stick its tongue in and get the nectar. Our Bombus affinis is at a competitive disadvantage, and so it has evolved the hole biting behavior, or nectar robbing behavior, where it bites a hole with its short tongue and it can rob the nectar. It's not making contact with male and female reproductive organs. So here, Bombus affinis is not a pollinator. In fact, Bombus affinis is a parasite. It is stealing nectar. It is not giving anything to the plant in terms of reproductive services. And in fact, what it's doing by stealing the nectar is it's deterring the good pollinator, right? Our Bombus viridis, if there's no nectar, as I mentioned before, Bombus viridis is just going to ignore this plant, species, or individual. And so our Bombus affinis is detrimental. So what's interesting is that Bombus affinis is the poster child for a quote unquote pollinator decline, even though with tubular flowers, it's not a good pollinator at all, if it pollinates at all. And we cannot call our Bombus affinis a pollinator until we give it a plant that it pollinates. So there's no, this isn't if we remove Bombus affinis from the equation, we can't call this a good pollinator plant. What does that mean? I guess it implies that at some point in the future, the animal that we're feeding is going to pollinate something, but unless you have that plant available, it's of no use, right? You're just feeding the animals. And so I could have hundreds of Bombus affinis all over obedient plant, and I'll be keeping Bombus affinis alive, but our plant's going to go down, right? Because it's not getting pollinated. Let's say it depends on seed production. It's getting no seed production because Bombus affinis is taking all the nectar, and there's going to be a consequence. Similarly, I could introduce a feeder here, and I could feed Bombus viridis and Bombus affinis and get hundreds and hundreds of Bombus viridis and Bombus affinis on my feeder. But unless I introduce my obedient plant, I have flower visitors. I don't have any pollination. I have no pollinators. So this is a critical point as we move forward, in that you cannot assume that because you see something visiting a flower that it is a pollinator. And also, this is the difference I was talking about just saving the bees. If we want to just save the bees, we can put up feeders, we can give them non-native plants, we can do whatever we want, but we can't call them pollinators, or we could call them pollinators, but in terms of their importance to native biodiversity, we have completely lost it unless we focus on the native plants that are being pollinated and the diversity of those native plants. So let's look at a real life example. Here we have the Pineland golden trumpet, right? So it's a tubular flower. The animals, if you have a long tongue, you can just put your head in, and the nectar is located at the base. Here are all the visitors, right? We've got different bees that are visiting. We've got some butterflies that are visiting. So all these species. The ones I've circled are the only ones transferring pollen. So we could see tons of things buzzing around our flowers and think that everything is great. But unless we know we have the pollinators, everything isn't great. We don't have that connection. We don't call them pollinators until we're pretty confident that they're making contact with the male and female reproductive organs. So if we removed our 3 species, we would have all these species visiting, but no pollination going on. All right. So just another example of this difference between flowers and flower visitors and pollinators. Now things get a bit more complicated if we look at natural plant communities, right? So natural plant communities tend to have multiple things in bloom at the same time. And so if we have our animal coming in to forage looking for food, let's say it decides that purple is the best, offers the most reward, and so it's going to stick purple, purple, purple. From the plant's perspective, that's ideal, right? The pollen is being transferred to individuals, all individuals within the same species. But let's say that our animal decides that, well, this one's good, but these ones are equally good. So I'm going to go from purple to pink to yellow, back to the purple one again. From the plant's perspective, this is a terrible pollinator. Pollen is being transferred between species. So this is a problem for 2 reasons. First of all, the pollen of this species is effectively being wasted, right? The plant can't mate with another species. And also, when this one receives pollen from another species, it blocks the surface of the stigma and prevents it from receiving its own pollen. And so it can't, even if it gets its own pollen, it's not going to lead to fertilization and seed set. So we don't have a pollination event. And so what the plants are doing, obviously there's strong selection on the plant side to get their animals that are functioning as pollinators, their best pollinators, target them and get them to keep moving among plants of the same species. And at the same time, our plants are trying to deter flower visitors, deter Bombus affinis, if you're a tubular flower, or anything that's robbing or is acting as a bad pollinator. So let's say it's still transferring pollen, but not as much as another species. It's going to target and fit, quote unquote, with that species that's the most effective pollinator, and try to discourage the ones that aren't that great. All right. All of this is going to translate into seed and fruit production, which is going to contribute to the population size of each of these plant species. On the animal side, the amount of nectar and pollen they collect is going to, so how well, and remember, we've got a lot of things, remember that first slide, a lot of different things, hundreds of individuals from the same and different species that are competing for a limited resource. The ones that are the best able to collect the nectar and pollen, they're going to be able to have greater reproductive success, and that's going to affect their population size. So if you're not a good competitor, right, then you're going to lose out and your population is going to go down. You can think of it as scramble competition for a limited resource. On the plant side, there are lots of animals to choose from, and they're trying to target their best ones and get rid of the other ones. So people think of plant pollinator interactions as this mutualism where everybody's happy, the plant gets reproduces and the animal gets food, but it's conflict. It's conflict, and this conflict is the reason that we have floral diversity. It's the reason that flower displays differ in size, shape, and color, and they're also the reason we have so many animals that are feeding on flowers for nectar and pollen. So if we were to look at, notice here I'm using the term animal pollinated plant, and all of its pollinators collectively, that is what's known as a pollination system. So again, the term pollinator implies a plant is involved, but nobody ever considers the plant. And all the efforts going on, it's put in plants to feed the animals. It's not, hey, these plants are in trouble too. These plants need to be pollinated, and we need to think about the pollination and as a system. Because as we'll see, without the plant, the functional role is lost, and the importance to ecosystems and biodiversity is completely lost. So if we look at these pollination systems, globally, there are about 300,000 or give or take, a few thousand animal pollinated plant species. And there are, couple 100,000 animals that that can function as pollinators, but are visiting flowers for using flowers as a source of food. And so there isn't a single animal on this side that can pollinate everything over here, and there isn't a single plant on this side that can feed everything over here. And what we see are subsets. There are a subset of plant species that are pollinated by a subset of animal species, right? And that's what we're referring to as these pollination systems. So to give you an example, here is what we see then if we look for patterns is we see that the flowers fit their best pollinator. It makes perfect sense, right? Because the plants have a lot, in terms of reproduction, and it's the same for the animal in terms of getting food for reproduction. So here we have our bumblebee that's visiting penstemon flower. You can see the male and female reproductive organs up here, but what we can see is the females up here, we can see that the pollen is on the thorax of the bee. Why? Because it's very difficult for the bees to groom pollen off of from this location. So the bees are using pollen. Pollen's a source of protein. For bees, without pollen, they can't make new bees, if you want to think about it that way. Nectar is a source of fuel. So you can see that this bee is using this flower for nectar. You can see its tongue's already out. It's collecting pollen and transferring it as a byproduct of the nectar collecting process. It will groom the excess to its back legs and take it back, drop it off at the nest. But from the plant's perspective, they want to minimize the amount of pollen that the bee's stealing. And so they try to put it in a place where the bee can't get to it, and that's the thorax. A better example, or another example, is in a hummingbird pollination system. The nectar is located down here. We see that here's the pollen. Where is it being transferred? To the forehead of the hummingbird. Why? Because if the plant transfers it to the wings of the hummingbird, there's not going to be very much. It's not a very efficient way to transfer your pollen. The female part is located up here. So the nectar's down here. As the bird moves in, it's going to brush its forehead against the reproductive part, the female reproductive part of the flower, and depositing the pollen, and then pick up more pollen as it feeds, and then will leave and go to another plant. All right. So that's a hummingbird pollination system. Here we have a fly pollination system. Here's the nectar is located at the base of this long tube. The fly has a long tube. Now this is a highly specialized system, meaning that there's one plant and one animal species. Without this animal, this plant doesn't reproduce, and without this plant, this animal doesn't eat. Other systems are more generalized, where the plant may have 5, 6, 7 different types of pollinator, and they all, or one category representing multiple species. Some are more effective than others, right? But all contribute to pollination to some degree. Here we have our monarch butterfly. You can see the pollen's on its tongue as it's probing for nectar. It's transferring pollen in the process. So all of these are pollination systems. And remember I said it's beyond the buzz, it's more than just the buzz. What that means is that in this plant, bumblebees aren't important. Bees are not important in this, for this plant species. They're not the best pollinator. The hummingbirds are the best pollinator of this plant species. The bees will come in and get the nectar, but they're not pollinating in the process. So the plant's going to deter the bee. And how does the plant do that? It has dilute nectar that's more favorable to hummingbirds than bees. So not only does it have traits to help to attract or maintain their effective pollinators, these traits also deter less effective pollinators. There's this push and pull that goes on, and we need to remember that when we're putting in plants. We're put in plants thinking, oh, this is a great bumblebee plant. You'll get one bumblebee species, and the four that we're concerned about aren't going to visit the plant because they're not the most effective pollinators, and they're being deterred. All right, so we need to start shifting our thought, shifting our view to more of this. We cannot talk about the animals and the plants separately. They act as a unit, and there are different types of units. So to give you an example, here's a bumblebee, the bottle gentian, a bumblebee pollinated plant. And so in this bottle gentian, the petals are fused together. And so the bee has to pry open the petals, which this bee is doing. And if I had the sound turned up, you can hear it going zzz, zzz, zzz. It's trying to vibrate those petals open, and it's squirming, it's wiggling its way down in to get the nectar located at the base of the flower. The reason that this is a bumblebee pollinated plant is because nothing else has the strength to get into this flower, right? And you'll notice that this is a long tongue bumblebee pollinated plant because the nectar, and once it gets in, it needs a long tongue to get the nectar. So only about 30% of bumblebees actually find the nectar in these flowers. And so the question is, well, why is the plant effectively deterring or not making itself available to 70% of its effective pollinators? And the reason is because for those few bees that get into this flower, there's so much nectar, more than anything else that's available, they'll travel far and wide to get to another bottle gentian plant. So that's the way that the plant maximizes the pollen transfer efficiency. It doesn't want its bees to go from bottle gentian to other things and then back to bottle gentian. It wants it to go from plant to plant of the same species. And that's the mechanism that the plant uses to trick its pollinator or to manipulate its pollinator into maximizing its reproductive success. Okay. All right, so hopefully at this point, you understand the difference between a flower visitor, a pollinator, and a pollination system, and why when you talk about pollinator habitats or pollinator plants, that term really doesn't, those terms don't carry, they're really meaningless. That really we should be talking about animal pollinated native plants and their pollinators, and so we should be referring to them as pollination systems. I prefer pollination system because it makes you think about the plant, that the plant is the player, is equally important, that the animal and the plant play an equally important role, and that we have to think of them as a unit. So these units are being degraded at an alarming rate worldwide. So just looking at bumblebees, here's some data I collected for bumblebees in Massachusetts. So, going through museum specimens, looking at what was a relative abundance of different species. Here are all our bumblebee species down here. And going back to the same location and doing surveys through 2015. This is 19 data, but I've been doing it yearly, expanding the number of sites where I'm doing these surveys, and the same patterns holding across sites. And you'll see that, pre-2000s, the blue bars, there are a lot of species that are in decline. Here's our Bombus affinis, which is thought to be locally extinct in Massachusetts. and throughout most of its native range. Other species are still holding on. Bombus terricola is closely related to Aphanis. It's a hole biter as well. It's still there. Vagans is another target. It's in trouble, and so is Bombus fervitus over here. The same thing's going on with butterflies. Here are all the dots here, butterflies that are in trouble, that are in decline. And not surprisingly, we're seeing the same thing with our native plants. They once were abundant and now they're in decline. So I want to just briefly point out here that there is a difference between being rare. So some species, either from the plant or animal side, are rare, and they've always been rare, right? But there are other species that were abundant, like our Bombus aphanis was in Massachusetts, and where I grew up in Canada, was the top 3 most abundant species, right? It was everywhere. And then 5 years later, I couldn't find it anywhere. So there's a very rapid decline. That's what I mean when we talk about rare, declining, imperiled. It isn't that I'm not referring to species that are rare. And this, when we say rare New England plants, I think here we're mixing in the rare species with the species that are in decline. But I'm really focused on those species that have seen significant reductions in their range and abundance. The other thing I want to point out here is that you'll notice that, yes, there are some in declining, but notice in the case of bumblebees, these yellow bars are higher than the blue bars. What does that mean? Well, these species are more abundant than they were historically. It's the same for butterflies. There are many butterfly species that are increasing. I could have put up another figure for other bees other than bumblebees, so non-bombus. Same pattern, some are increasing, some are decreasing. And it's the same for our native plants. Some are increasing and some are decreasing. So the point here is that not everything is in trouble, right? Some species do not need our help. That whatever we're doing to drive others to extinction, some to extinction, others are thriving under the same conditions. And so we cannot just look at how many butterflies or bumblebees or other bees we see on our landscapes and say, okay, this is doing its job. We are helping pollination systems because I've got tons of bees on this plant and it must be helping. We'll see in a second that is misleading. To give you an example, Bombus impatiens, the common eastern bumblebee, at this time of the year, the numbers are starting to pick up. By the end of August, I'll go to a site and see over 2,000 individuals. All of them are Bombus impatiens with a couple of other species sprinkled in. At the same time, if we were to go to pre-2000, we would see 4, 5, 6 species in addition to Bombus impatiens, right? There were other species, that Bombus vagans being one, that was equally, if not more abundant in many areas. So, so this difference between abundance and diversity, I'll talk a little bit more about in a second. So why should we care? Since I'm the 12th talk on pollinators, I'm sure you're all, you've all received this message about the importance of pollinators. There are 2 reasons that pollination is important. One, this particular one is getting a tremendous amount of attention and has since we knew that bees and other things were in trouble in the early 2000s. Interestingly, the public became aware of the decline with the colony collapse disorder in honeybees. So honeybees are shown here. They're a non-native species. But we knew that Bombus aphanis and other native bees were declining in the late 90s, early 2000s. So well before I would, give these talks and people would say, oh, you should tell people about this. And I would, and it really didn't matter. So I'll say, that the honeybee, in terms of bringing it to the public's attention, the issue and the importance of pollination has done a great job. But there are a few things I want to point out. So honeybees, as I said, are non-native. We have a couple of native species that function as pollinators of crop plants, right? So we've got a handful of native species. We've got our honeybees, and they're pollinating a handful of plants, right? And those plants are not native plants. They may have been native at one point, but here's what they have to deal with, right? We've got acres and acres and acres of one thing in bloom for a couple of weeks. Well, to get all this pollinating done, we need a lot of individuals. Well, each one of these honeybee hives has, 10 to 30,000 individuals, and we can saturate the system and get all this pollinating done. So the honeybee is ideal for this type of situation, and certainly it plays a tremendously important role here. And to a lesser extent, I guess you could look at the honey business. But we've got a handful of species here. We've got a handful of species here. And the products of pollination in this context are, but there's one species that benefits. Well, we could throw in domesticated animals as well, but we're the main species that benefits from pollination in an agricultural context. And obviously we're important, and this plays a tremendously important role. One thing I want to point out is that all that matters here is the abundance of bees. Bees are doing the pollinating here, and in particular, honeybees are playing an important role. Certainly, native bees, wild bees can contribute to pollination to some extent, but other things aren't doing the pollinating. They're not, they're not good sources of nectar and pollen, but when it's the only thing available, I use this analogy all the time that you could think of plants as fast food or fine dining. This is fast food. The animals aren't going to starve, and so they'll eat it, but it certainly isn't, they're not necessarily benefiting from it. The one thing I want to point out is that, we have thousands of bee species native to North America. Only about 5% of them are visiting crop plants. And guess what? The ones that are at risk and declining because their numbers are so low, they're not major pollinators of any of this. And so and this is where the problem comes in, when we try to help the honeybees and we try to conserve pollination in agricultural context, we're really focused on abundance. Just get a whole bunch of something there. It doesn't matter what it is. And it'll do the job. When we switch over to the second, and what I think has been neglected, although things are changing, which is great to see. When I started giving these talks, most people weren't aware of this. Now more becoming aware and realizing how important it is. But in the ecological context, here the importance is not, again, the bees were important because of the crop plants being pollinated. And you can think of that as an artificial pollination system, I guess. Here, what's important are the connections between native plant and pollinator species, those pollination systems, and importantly, the diversity of those systems. Why? Because again, what I said in the agricultural context, we've got a handful of animals pollinating a handful of plants, feeding one species. Here, we have hundreds of animals pollinating hundreds of plants that are providing food, shelter, and nest site for hundreds of species, right? The seeds and fruit produced are feeding birds and small mammals, which themselves are food for predatory species. And so our diversity here, the different types of food, the different host plants that we provide, the different types of plant material for nest sites and shelter is supporting diversity on this level. So as we start to degrade this, so that's why they're referred to as keystone interactions, right? Saving the bees by themselves, that's one species you're helping. Saving the bee and the plant that it pollinates, that's saving two species, plus all of the animals that wildlife species that benefit from that interaction, those products of pollination. And so as we start to degrade this, we're going to start to see effects at this level. Less food here, which means less food for our owls. Owls are in trouble. They love to eat voles. What are voles eating? They're eating seeds and fruit. Where are those seeds and fruit coming from? They're coming from a pollination system. Right? So we'll get to the point where we degrade this so much that we're going to start to see a massive reduction in biodiversity and what's called ecosystem collapse. And this is a major problem. Now, in the agricultural context, it's a problem because of food on our table, 1 out of 3 bites of food. I'm sure you've heard that number thrown around, and also agribusiness. But the reason that this is important in an ecological context is because of what are called ecosystem services. These are things we get from nature for free. Pollination is one in an agricultural context, but we've got carbon sequestration, water purification, soil decomposition. All of these things are things we get for free. And if we lose diversity and ecosystem functioning, we are going to, we lose the diversity, we're going to lose these ecosystem services. And I'll tell you that, if there's a significant price tag associated if we lose them. And as they say, extinction's forever, we're not getting these services back. And if we can invent something to replace it, great, but it'll come at a great cost. And so even though there isn't an immediate economic incentive for conserving these native plant pollinator systems, long-term, there definitely are going to be some costs, and it's going to affect human health and a variety of other things. So it's very important that we maintain this diversity, that we can think of these systems as the foundation of ecosystems, right? And we're going to what we're going to do is the ones that are in trouble, we're going to help them out so we can maintain our foundation, so that our foundation doesn't get degraded to the point where we get the house collapsing type thing. And just to give you an example here, we've got our owls that are relying, as I mentioned, we're feeding on small mammals. Those small mammal populations depend on the availability of food, right? Where's that food coming from? It's coming from our pollination system. Same with seed-eating birds and birds that eat those seed-eating birds. So we need to realize that everything is connected, and at the center of this connection, we have our native plants, right? Again, the native plants are able to perform their function because animals are pollinating them, keeping plant populations going and keeping the food supply going to all these other things. And so, and again, that hopefully I'm hammering this, I'm hammering you with this point over and over again, that it's the native plant and the animals that pollinate it that's important, not just the animal or the plant by itself. So what's causing the problem? Here are the proposed factors. My lab's studying all of these things. I could give a whole other talk on effects of pesticides and disease. These two things are our problem in agricultural and urban areas where we're using pesticides. The take home is, don't use them. And, here there are some diseases moving from honeybees to bumblebees, or sorry, honeybees to native bees. And so we want to keep the two worlds separate, although the two can coexist. And I'll get to that near the end. Climate change certainly is playing a significant role. If you think about the overwintering of certain species, they have to drop below a certain temperature, or if they do drop too far down, they're not going to be able to survive the winter. It's putting plants and pollinators out of sync with one another. And so these are all important things, but I don't have enough time to focus on them all. And so what I'm going to focus on is what is, I'm sure everyone that's in this game would agree that habitat change, habitat loss is the major driver, and these other things certainly don't help. They're helping to accelerate declines in these areas. So by habitat change, it might be something, it might be removing good nesting floral resources. It may be the introduction of exotic species. So by swapping out or planting non-native or exotic plants versus native plants, that's disrupting those connections that I talked about. Because the reason that those plants are here is because they're connected in an important way to everything else. So when we try to replace them with things that the animals using the plants have co-evolved with, it just doesn't work. It may work, but it doesn't work. It may work in some cases, but it certainly, even in those cases, isn't working as well as it does, because that's the way that selection has made things, right? It's that we have diversity on both the plant and the animal side for a reason, because they go well together, or they fit well together. So how is it that we change and restore these connections? This is where I start to get into the Beecology project. And I apologize if you thought that the introductory material was too long, but I really feel it's important to understand what I'm doing and why what I'm doing is different than what everybody else is doing. And I hope that will change. We really need to understand some of the points that I brought up in the first part of the talk. And to drive one of those points home again, this idea that we need to feed flower visitors versus conserving these pollination systems. We use a hummingbird example this time. We've got our hummingbird feeder, and we've got our hummingbirds. It has a choice. Should it visit native plants, or should it visit the feeder? Right? The choice is fairly obvious. That we've got unlimited food source, one-stop shopping here. We've got to visit hundreds of flowers here. It takes us more time, exposes us to predators. So the birds are all going to go over here. So we may be able to support many, many male and female hummingbirds. They can raise their young. So we're supporting the animals, but who's pollinating the plants that are depending on hummingbirds for pollination? Nobody. So this is breaking the connection, right? We are breaking the connection by pulling our hummingbirds over here. And these plants, these plants are going to, they can't maintain those connections as well. So it's far better to plant Monarda, right, or lobelia, or get some jewelweed on your property, and it doesn't take much to restore those connections versus putting up a feeder. And the other thing is we could replace our feeder with a European honeysuckle, which hummingbirds love. Here we're feeding the birds, but again, the pollination products and the plants aren't going to be used by the things that need it. So really it's the same thing as putting up a feeder. We're breaking the connection. And what we want to do is give the animals what they want, what they prefer, and that's the native plants, and restore their functional role as pollinators. So the one thing that when we're talking about habitat restoration that is important is that we get rid of this one size fits all, that this is a good bumblebee plant, this is good for small bees, and this is a great butterfly plant. Each species has a specific set of habitat needs. And the reason that we have so much diversity is because they have unique needs. If they had the same needs, everybody would compete with one another. There'd be one species standing. So we need to appreciate that each species is different, and we need to figure out what those differences are at each life stage. And this is what I refer to as an ecological preference. So in butterflies, they need a host plant and they need a nectar plant. And I throw the monarch up here because it's easy. It needs milkweed. No milkweed means no monarch larvae have nothing to eat, and that's obviously going to affect monarch populations. But that's true for all butterflies. They need their host plants. If they don't have their host plants available, then you can have all of the flowering plants that you want, your butterfly gardens. You can have them, you're feeding the adults, but there won't be any adults there because you didn't get them through the larval stage. So you need to plant both of these things together. You need, and with bees, you know, they need overwintering sites. When they come out in the spring, they need to find nest sites. Each species has different nesting preferences. They have different overwintering preferences. They need nectar and pollen. As I mentioned, the nectar is a source of fuel. The pollen is a source of protein. If they don't get those enough pollen, the right pollen that they like, they can't make new bees. The population is going to start to be affected. Then they mate, and then they overwinter. So it's the same general cycle, but they have different needs at different times. And so what my research focus is then is to try to figure out what the species at risk need. There are a lot of information out there telling you what they need, but as we'll see, it turns out that most of what's out there is not what they prefer to eat and not what they need. And so the other question that I'm starting to ask, and again, it's, as I said, it's about the pollination system, is who are they pollinating? These bees that are in decline, the long-tongued bees, they pair up with tubular flowers, not surprisingly. And if they're the most effective pollinator of tubular flowers and those plant species are in decline, then they might be in decline because their most effective pollinator is also in decline. So if we want to understand how we're going to restore biodiversity, we really need to understand what the needs of the individual players are. And so the first thing we need to think about when what do at-risk species need is we need to know when are they active, right? Again, this idea that even in bumblebees, not all species are active at the same time. Some species are finishing their cycle and we won't see them for the rest of the season. Others, as I mentioned, are starting to ramp up their numbers. And so we want to make sure that we know when those at-risk species actually need our help. And so here are a couple of butterfly species. You can see when their flight times are. They're most active in this period. So from late June through into early August, but mostly in July. And this one over here is more concentrated in July as well. So we want to make sure that we have host plants, but the nectar plants are available when these adults are around. Here's a Andrena bee that's in decline. And notice that it's done, so you're probably hard to see, but this is March, April, May. By the end of May, this species isn't around. So if we're planting to help the bees, quote unquote, in June, we have things that bloom in June and July, this species is already done for. It's already done its cycle. So that's why we need to think about, okay, well, what's blooming in April, May? And guess what? It's willow. So willow is supporting this species. And if we don't have any willow that's blooming or anything that's blooming in the spring, then we're not helping. So we really need to know when things are active. And also here are the bloom times for all these are native plants. We can see that, you know, obviously if we're planting aster, it's not going to bloom until September. And if we have, what's going to get our butterflies or bees or whatever to be able to pollinate this in September? We need to make sure that we have what the species needs throughout its cycle. Our bumblebees are active right when the snow melts all the way till the first hard frost. So we need to give them nectar and pollen sources all the way through versus our Andrena, which we have to make sure that we load up what it likes in the spring, okay? And so as I mentioned, willow turns out that willow is extremely important. Planting a couple, and willow comes, there are many willow species, and willow is critical to have in the spring. So just by planting willow, you're helping all the threatened bees. They're out in the spring. All these threatened butterfly species are flying around in the spring looking for nectar. The bees are looking for pollen, so you want to plant male willow in addition to female. And some butterfly species at risk are using it as a host plant. So just one willow, right? A couple of willow plants, you're having this positive impact for the species that need it. Right? So it's just, you know, thinking more about what goes in and what exactly are you trying to accomplish with your planting goes a long way. So we have to think, again, I mentioned very early in the talk that we need to think about things from the perspective of the animal. The German word for this is Umwelt. And my German's terrible, so I apologize. But if we're a short-tongued bee, obviously we're not going to be able to access tubular flowers. So if we look at our bumblebee here, medium tongue, but this is Bombus impatiens on goldenrod, you can see its tongue. It just has to walk on the top and the nectar is exposed. It's very easy for short tongue bees and also butterflies that are able to probe around to get nectar. And that's why we see wasps and flies. That's why we see a ton of things on our goldenrod, right? But our species at risk, our bumblebee species at risk, all have long tongues, and they're not going to touch goldenrod. Why? Because they can't compete for nectar because their tongue gets in the way. So these species, what they want, we have long tongue bees, they fit with tubular flowers. So here's jewelweed. It's got the nectar is located at the base of the spur. This is our Bombus fervitus visiting the jewelweed. And our short tongue bees can't get their tongue to where the nectar is. And so they are not going to visit. They're going to be staying on the goldenrod, but our species at risk are going to be over here. For our Bombus terricola, I mentioned it's a hole biter. It's going to be exploiting the system and feeding, and then it would come over here potentially with its short tongue and collect nectar here. So it could sort of serve a dual function. But for these two species, it's all about the jewelweed and other things that have long tubes. So we need to consider you know, and not surprisingly, these long-tongued bees are the main pollinators of these plants, and the short to medium-tongued bees would be the main pollinators over here. The problem is that when I mentioned this one-size-fits-all strategy, and the idea that we're really just feeding the flower visitors, we're not conserving pollination, the function, like pollination as a process, and the ecological role that the pollination plays. So this is a list. You know, I could have pulled a dozen lists to show you, but notice here that this is for bumblebees. So the idea, digger bees, everything is just grouped. And I guess you go down the list and you just pick a couple and you put them in and then you're helping. The problem is that many of these aren't native. So if you're putting in a non-native plant, you're not restoring the connection. A lot of these are rarely visited or infrequently visited by bumblebees. And if we look at those long-tongued bees that are at risk, clover is the reason that we haven't lost Bombus fervitus is because of red clover and vetch. And because the native plants that they prefer aren't on the landscapes. It's all old field landscapes where they like to nest, and it's all vetch and red clover. This red clover is not even checked here. And then, you know, Penstemon's a great one. It's highly preferred. So this is a keeper, definitely. But some of these other ones, you know, common species love it, but the species at risk don't. And rose is a #1 pollen source, but it's treated as though it's equal to all these others when really it's not. The importance of rose and Penstemon blows everything else here out of the water, even the other natives. And so, you know, this one size fits all approach, we need to shift it if we want to save anything. Even if we just want to save the bees, we have to move away from this, you know, just save the bees, quote unquote, forget about the pollination part. We need to think more about what we're putting in. And so here are a couple of examples of seed mixes. You know, we got non-natives. Notice that these flowers look the same. I told you that we need tubular flowers. I don't see a tubular flower here. Why? Because this is for honeybees. Honeybees have a short tongue. They don't care if it's native or non-native because they're not pollinators of native plants. There's no native plant that depends on honeybees. So you could put in whatever, and this is feeding our honeybees, but it's not doing anything ecological. Here, there are a couple species, the Monarda and the lupin's good, maybe the Liatris. But again, a lot of this, its composites are no good pollen sources here. And the lupin and the Monarda are good for a couple of weeks out of the year, but what are they supposed to do for the rest of the time? All right, so we really need to shift from feeding common species to helping pollination systems at risk. So how is it that we figure out what they want to eat? I've conducted weekly surveys across Massachusetts and other places from March to October, looking at what are the bees visiting? Are they visiting for nectar and pollen? And just compiling data. Is it a male, worker, or queen in the case of bumblebees? But importantly, what I've done, I can't get everywhere in the state. I noticed some patterns early on. There's no way I had the person power to be able to collect enough data to really figure out what they need, to see if the patterns that I found at a couple of locations were consistent across the region. And so what I did was I developed the Citizen Science Beecology project. You can get more information on the project here. And then I also have information on my lab website. And so there are 3 goals to the project, right? Again, there are a lot of projects out there to monitor bee populations and where they are. And although that's an important component of Beecology, I definitely want to know where they are. What's extremely important to me is what are they doing? What are they visiting? Are they visiting it for nectar or pollen? Because, and where are they nesting? And where are they overwintering? This is the ecological data. And so, phase 1 of the project was to collect ecological data on the foraging behavior of bumblebee species at risk. And so through crowdsourcing, I can get enough data, look for the patterns, and then develop a plant list that caters to the species that need it. The second important part of this Beecology project, if you're interested in participating, is that it is extremely important to me that you see that you made a difference, right? I can tell you what plants to put in, but I want you to see that the species at risk weren't there. And when you put in the plants that I suggest, they are there. So you are able to assess the quality of your habitat, make changes, and then reassess to see that you've made an improvement. And I offer a lot of workshops. I gave 2 workshops last weekend, Saturday and Sunday, on how to tell these at-risk species, how to assess floral resource quality, and so I'm happy to do that. I don't have enough time to go over all of that today, but just that is an important goal of my Beecology project, empowering citizen scientists so they know that what to do and what to look for. The third thing is to the Beecologists do is to help to use the data that they've collected, and we're at this phase now, to restore and protect native pollination systems. Get the plants that these species prefer and get them in the ground so that we can help to maintain the populations, and then over time increase the populations and expand their ranges back to where they were historically. And so to help the data collection part, we have a web app. So if you go to the beecology.wpi.edu website, there's a web app. And so the key here is just go out and take a short video, 10-second video of a bumblebee and now butterflies. We've just added butterflies to the app, so you can upload butterfly videos, and we're working on the ID, trying to work with iNaturalist to use their API to get it into our app so that we can help with the plant ID and help with the butterfly ID. But currently you can upload your bumblebee videos. It'll help you to ID the bumblebee, the plant species, and importantly, the behavior, where they're collecting nectar and pollen, and then get that to our database. We have a lot of, if you go to the Beecology website, there are a lot of tools, visualization tools. You can see the plants that the different species prefer based on the Beecology data. We're developing high school curriculum, hypothesis testing, and a bunch of things around the Beecology website. So there are a lot of interesting things there, and we continue to add things. I have weekly meetings to go over. If you have any suggestions, feel free to email me, and we can work on adding them to either functionality to the app, coming out with a new comments function, so you can tell me what you think it is, and I can communicate with you back. I can say, well, I changed the ID because it's actually this species for this reason. And this is in collaboration with some computer scientists at WPI, and we have a whole team of students on the tech side of things that are helping to develop the project. And so if we look at my own data, right, so we're looking at nectar preferences. I'm collecting all of these data. Here are results from July. And so here are our bumblebee species. I've separated them out to long, medium, and short tongue species. And not surprisingly, tongue length matches up with tube length in the plants, right? So our long tongue bees are all on vetch and red clover. Our short tongue bees are on milkweed and goldenrod, which makes sense. But what I want to point out here is that you'll notice that there's variation. Even though these bees all have long tongues, Borealis is preferring milkweed, and our vagans, which is in trouble, is preferring bush honeysuckle, loves bush honeysuckle. So there were 25 plant species where I saw at least one bumblebee, right? But you can see here that for every species, the vast majority of their visits were on just 2 species. More than 50% of the observations were on 2 species. Clearly, they have a preference for certain flower types. Some of it can be explained by tongue length, but a lot of it can't, right? Here are 3 medium tongues, same bee size, they're all out at the same time. All these bees are present at the same time, and all the plants were present at the same time. This Bombus impatiens, the common eastern bumblebee, guess what it likes? Purple loosestrife and vetch. So Bombus impatiens and honeybees love purple loosestrife. You'll see thousands of bees. It's all mostly impatiens, definitely common species, not the species at risk. When we look at pollen plants, remember they need pollen to make new bees. Again, 25 plant species where we saw a bee on a flower, only 2 species were they collecting pollen, actively collecting pollen. And that was meadowsweet here, white meadowsweet, and the St. John's wort. And this was the non-native St. John's wort at the site because it was an old field site. But what this tells us is that although they're very specific in their, or certainly more specific in their nectar requirements, pollen, they're using the same thing. There's a lot of overlap. Overlap means competition. And so if we don't have a lot of meadowsweet, we're going to have winners and losers. And guess who's going to win? The common species in the hundreds, not the 1 or 2 species that are struggling. And so this was another very important finding. And what's interesting is if we look now, let's look at the Beecology data for July. And I'm sorry that the text is a bit smaller, but here are 3 species, Bombus fervidus, terricola, and vagans. And what we can see is we can go down the list, and here's the monarda, that Bombus vagans likes the monarda, self-heal, red clover, and the cowvetch, completely consistent with what I was finding with more rigorous surveys and knowing how much resources there. And so I've been using the Beecology data to test the patterns that I've been seeing. It also helps me to locate where these species at risk are, so it can go and survey those areas to look for new connections, and also identify areas where the species used to be, where I think we need to do a lot of work, where we need to put in the plants that it prefers. But collectively, these data have gone into my plant list, which is available on my website. So this is targeting bumblebees. But again, Beecology is not just about the bees. What I've done is gone to the literature, found the butterfly species at risk and the other bee species at risk, looked through Discover Life and published sources on what it is that they're visiting, when are they active, and so what would good matches be. We certainly need more data on the butterflies and other bees in terms of floral preferences, but there's enough there that now you can go to the list and you can say, okay, in the spring I'm going to take here's my willow, and it's helping out the bees, and so this is for pollen sources, good pollen sources in the spring. Willow is a great pollen source in the spring. And just go down the list and you can find out what you would need for a particular time, and it's got soil conditions, whether you've got full shade. The hairy woodmint is great for full shade if you have it, things like that. And so to give you an example then of how we reconnect, remember I showed you the list before that had non-natives. This is targeting the species at risk. So just by putting what's on the slide in your, putting these species in, you can effectively at least maintain, and depending on the number of these species you have, you can increase numbers of these threatened species. So in the spring, lupin and early spring, you can throw willow in here. I have willow more on the pollen side, but lupin and wood betony are 2 native plants that are great for queens in the spring. Moving into Penstemon Hercetus, the hairy beard tongue, and Penstemon digitalis. Hercetus blooms first, digitalis second. It's a great combo for these species. Into the monardas, Prunella vulgaris is a great lawn replacement plant that they love, and then into jewelweed in the fall. Purple giant hyssop should also be here, or could swap in for, it comes after the monarda and before the jewelweed. In terms of pollen sources, native roses, into the meadowsweet. Purple flowering raspberry is tremendous pollen source. This is the 4th week that my purple flowering raspberry has been blooming, and in terms of pollen, it's incredible what's on that plant in terms of bees collecting pollen. Okay, so we've got the list. That's how we came up with the list. Everything that I do is data driven. If a new plant species, if I get data on a new native plant species and a new connection, it goes on the list. The list has changed considerably since I gave this talk in 2018. It's improved, and now I've got enough data where I'm very confident that these species will help to reconnect. So we're reconnecting these animals to the plants that they pollinate, and I have a grad student that's looking at each one of these species to see how effective these species are as pollinators versus more common species. All right, so when I say native or species, again, thinking about things in terms of ecotype, straight species I'm talking about. I'm not talking about cultivars. There is a huge problem with cultivars. Even though we think they're the same, the animals do not think they're the same more often than not. And so Annie White at the University of Vermont studied this, has some great visuals. And I'm sure I'm not the only one that has noticed the problem with cultivars since grad school. I've been burned by cultivars many times. The problem is when you breed for show, breed for color, breed for size, cold tolerance, something's got to give. The plant only has so many resources to distribute to different things. So when you breed for one thing, the first thing that goes is the nectar. And this is because genetically nectar is linked to a lot of these traits. So when you change one trait, you're changing the nectar as well. And so she looked at how well these plants supported. She has not looked at pollination, and I have a student that's looking at the pollination side of this. But we can see that here's each of these bars corresponds to the different cultivar color. You can see that the Echinacea, the straight species, is preferred, and all the others are not preferred by varying degrees. So here's our bumblebees. They don't really even touch the pink one, the yellow one, not so much. All right. So they don't, and as I said, you're changing things. You might change the chemistry of the plant. So it's no longer a suitable host plant because the thing that's using it as a host plant, the larvae don't like to eat it, or the female doesn't want to lay eggs on it because it doesn't have the right chemistry. It could be any number of things. And in addition, the seeds may be off, so the birds don't like the taste of the seeds because of the chemistry. So there are a lot of things that could go wrong. It's just better to go for the straight species. And so to give you an example, I mentioned the nectar. Here's lobelia, here's a hybrid cultivar. They look the same to us, they look great, but they don't look great to the hummingbirds, right? Here's the nectar and in the straight species, and here's the nectar in the hybrid. Our bird's gonna come in and say, not enough nectar, and move to something else. And so even if it did produce, the pollination is going to be affected, assuming that the seeds were right, the birds aren't gonna do the pollinating. And so it's just, again, it's breaking that connection that I talked about or have been talking about. So what can we do to help restore these systems? Again, the Beecology project, this is goal 3. We're going to help to restore and protect and assess the habitat that we have. Think critically about the plants that are there, the plants that aren't there, and what we're trying to do. I always talk about goals. If your goal is to have a showy ornamental garden, great. If your goal is to have a vegetable gardens, provide vegetables, great. I have no problem with that. If your goal is conservation, you need to think about what you're doing. And a lot of what people say you should do isn't really helping the way that it should. And so again, you just need to think more about what's going in and what impact it's having. So the first thing you should do then is take an inventory. Are you feeding animals, or are you restoring, do you have your pollination system? So you may have these species at risk, but if you've got vetch and red clover, what we want to do is think about what can we swap them with. I'm not saying to go in and get rid of all the non-natives because that's how they're surviving. Introduce the natives. When the populations get to what the non-natives are, then we get rid of the non-natives and we can restore the connection. We may have native plants present, but we don't have these species. Why? Because maybe we don't have things for them to eat earlier in the season. So we want to think about how we're going to get them to our native plants, depending on when our native plants are blooming. We want to identify our targets. As I mentioned, I've got a handout I'll give you to ID the bumblebee species at risk. There's a lot of information on butterflies we're putting together and other bees, but it's very important that you know what a Bombus fervidus looks like to know if you have them, and if you don't, if you put in species, whether you now are seeing them. And, I live in an urban area. I put these plants in. I've never seen a Bombus vagans. Today I saw 4 or 5 alone. It really is that rapid and that dramatic of a change. If they're in the area, and if they're not, again, if more people do this, we expand, we're able to pull them into new areas, and you'll see them. And I certainly in Southborough, where I work a lot, people have put in plants and didn't see them, and then started to see them as the plants start to establish and they increase in number, become a good resource. And so this is the handout that I'm happy to give. It identifies the species at risk, where they're located, high, low elevation for New Hampshire. You're going to start to see Terricola if you start to go north and more to the west. In Massachusetts, you're not going to see this unless you get above 1,000 feet and farther to the west. And so things are going to change a little bit, but more or less, I think you could use this guide in New Hampshire and states, surrounding Massachusetts. This is something, again, the flipping things to the plant side. What we want to do is go out and document. Do we have a native, non-native cultivar? Of the native plant species do we have? What is the flower shape? Do we have a lot of composites? Do we have tubular flowers throughout the season? So check off how many of these things you have and think about who you're helping and who you're not helping with the plants that you're putting in and when they're blooming. So it's really, when I say inventory, Excel sheet, you become the scientist. You figure out what you have, do a pre and post survey, that type of thing, to see that you've made a change. The other thing is, the question is, should I add native plants or should I just remove exotics, and particularly invasives? So here, Breakneck Hills, my first study site in Southborough, 40 acres plus, covered with, I said vetch red clover, a lot of natives in here. We're working on increasing those numbers. But down here is a wet meadow covered with purple loosestrife. We remove the purple. So the purple loosestrife, lots of honeybees, as I mentioned, lots of the common eastern bumblebee all over here, some of the other common bumblebees, the odd butterfly certainly, but none of the species at risk, right? So what we did was took out the purple loosestrife. 3 years after taking out the purple loosestrife, what showed up? Tons of Mimulus, monkey flower, native, all over the place. And guess who started showing up on site? This is Bombus. The first one was Bombus vagans. This is Bombus fervitus, the species at risk. I've been surveying breakneck since 2015, tend to see 1 or 2 individuals. Last year with the Mimulus, I saw, 20, between 10 and 20 individuals in one survey session. And these are all the other species that came up just by removing a little bit of purple loosestrife. 1/4 acre of purple loosestrife makes a huge difference, in addition to putting in the Beecology habitat, which I'll talk about in a second. So removing invasives, I've seen this other places as well. You pull them back, you'd be surprised that the natives that are there being suppressed, they're just waiting to get the chance to come up and repopulate areas. And so if we want to add plants and we don't have a lot of space or money, let's plant high impact plants. So native roses, as I mentioned, good pollen sources. They're nectar, or they're host plants for apple sphinx moth. Apple sphinx moth we can think of as food for eastern whippoorwills, which are a species at risk. The rose hips are going to feed all of these bird species. And so just by putting in one plant, you've got a major impact on multiple species through again, the pollination and the existence, the presence of that plant itself. And it's not just about the flowers, right? Grasses, native grasses and sedges are just as important as our flowering plants. They're not as showy, but they're just as important. Why? Because first of all, they're host plants for many butterfly species at risk. Birds love the seeds, right? So we're feeding the birds, but also we are providing nest sites for rodents, maintaining rodent populations. I know rodents have a bad rap because of ticks and Lyme, but they're feeding our bird species at risk, like our owls. Importantly, putting things back in pollination system, the context of pollination systems, our bumblebees are using abandoned rodent nests in old field areas as nest sites. Bumblebees do not dig their own nests. They exploit existing cavities and structures and abandoned rodent nests. And so by having high rodent numbers, we're providing the nest sites for these bumblebees that need it. Bombus fervitus nests in grass areas, surface nester, using abandoned rodent nests. If we mow our grass, The rodents aren't going to nest, or if they do nest, they're going to nest below ground. Bombus fervidus isn't going to have a nest site available, and it's going to move on or not be present on the landscape, no matter if we put the plants in or not. So again, it's all about connections. Lastly, nesting habitat. Different bees have different preferences, and I'm sure other people have talked about this. So we've got, we want sandy soil exposed. We want pithy stems in our native plants, so that we can cater to help the cavity nesting bees. But for Bombus fervidus, the bee I was just talking about, bandit rodent nests in grassy areas, this is how much space you need to not mow. So you leave it for 1 year, let them find a nest site. This is showing you where the nest site actually was. And the reason that they didn't mow, they always mow this, is because of COVID. So one of the positives to come out of COVID was that there was a Bombus fervidus nest here that wouldn't have been here otherwise, and the numbers were really good. There were queens produced, and the numbers have increased this year. And we told them not to mow, or asked them not to mow, and they've left some space, and we saw 2 fervidus queens. And so, just that little bit's helping out. And so just finishing now the talk to talk about restoration and protection, success stories, I like to call it. So I put in sites in Dartmouth, Southborough, Northampton, Framingham, and putting in plants, not plugs, species from my list, those critical species last year. I have to thank the volunteers in Southborough and Dartmouth for helping me to get these plants in. I don't have a green thumb at all. So last year was really dry and hot. So there was a lot of watering and, problems with grasses, which is why they're wood chips. But I'll say that last year, as soon as these plants came into bloom, these are all pictures from the habitat. So again, 40 acres, I see 1 or 2 fervidus. These pictures are all fervidus within this small habitat within 40 acres. These ones are of Dartmouth. So we're able to pull in and support the species at risk. And this year, so late last year, showy goldenrod is a great late season, not just for the species at risk, but for everything. It's not like a typical goldenrod. Here is a Bombus vagans queen on our showy goldenrod. I've surveyed a ton of goldenrod and have not seen Bombus vagans queens foraging on goldenrod. This one has a bit of a deeper, I think it must produce more nectar. We're looking into exactly why, but showy goldenrod is a species in decline. And I had 3 sites and saw 3 vagans queens, separate sites. Dartmouth and Southborough are separated by a considerable distance. But the first time I've seen vagans queens on showy goldenrod or on a goldenrod. Here's Penstemon, hairy beard-tongue Penstemon Hercetus. This is in, this is Bombus vagans. This is from this spring. Over here, we've got a video. The bags are a pollination experiment that my student's doing to figure out who the major pollinators are, because this is another native plant species in trouble. This is Bombus fervidus. You can see Bombus fervidus in here, again, going right in, pollinating, and we're going to see how effective both of these are as pollinators compared to more common short-tongued species that were also observed visiting. So with that, I would like to thank everybody listed here. If you want to contribute to the project in any way, here are 2 people that have contributed funding-wise to the project. I really appreciate their efforts. They provide money to put the plants in at these different sites to help these species at risk. But if you want to take a video, you want, every little bit helps, and I appreciate all of the beecologists, no matter what their contribution. And so with that, I'll happy to take any questions. I would like to thank you for your time.
Wow, thanks, Rob. That was
You're welcome.
amazing. Here's a few questions. So I'm wondering about the 30 by 30 foot goal to preserve 30% of land by 2030. Are you or experts like you able to weigh in or work with those leaders choosing where to preserve or how to improve the habitats chosen by planting smart?
Right. So at this point, no. I would like to participate more. Interestingly, it's much more efficient for me to work through community and citizen scientists than it is through the government. I'm a part of many government groups, and they have their own way of doing things. And, what I'm doing doesn't seem to fit with what they want to do at this point. I hope, and this is, I'm speaking of Massachusetts, but I certainly am always willing to collaborate and participate and help out and contribute any way that I can.
So a couple of specific, how can you tell if plants you've had for a long time are straight species or cultivars?
Yeah, good question. So you really can't. I mean, you could, if you notice something different about, if you can get a hold of a straight species and do a visual comparison, other than that, it's hard to tell. I'll say that if you don't see anything, so the way that I can tell is, you know, things. So I got some obedient plant and nothing was visiting it. And I know that obedient plant pulls in long-tongue species. And not only with the long-tongue species not visiting it, nothing was visiting it. And so I pulled and did some nectar tests, you know, with the microcap, and no nectar. So I went back to the source and said, well, what's going on here? And it turns out that it was a cultivar, Miss Manners. I didn't know that. And so if you don't see anything on the plant, odds are something's up. If you do see what you're supposed to see, so if it is a plant on my list and you're not seeing what you should be seeing, it may have been changed in some way, but really it's difficult to tell without doing some sort of genetic analysis if there's no visual indicator.
So there are a couple of questions about good resources for native plants.
Yep.
I guess at least some individuals know about the Native Plant Trust, but are there other places to get resources on native plants?
Yes. So so for some things, Native Plant Trust is good, but for some of the harder to find plants, native plant species, so Northeast Pollinator Plants is great. It's in Vermont. For Prunella, that's the place to go. There are two species of, two subspecies, one's native, one's non-native. But I will say that blue stem natives just started up in May 21st, which is in Norwell, Massachusetts. And they do online ordering and I think ship. But they are going to, and made it a goal to have all the plant species on my list. So I'm working closely with them. They already have a lot of what's on my list, and they're working hard to get, for next year, they're going to have everything. So I'd say that's a good one. There is Wing and a Prayer Nursery, Amy Pulley, which is in Cummington, Massachusetts, that I use for plants, and then various other places. So if you have problems getting a plant species, I'm also, a lot of these habitats, I'm collecting seed and doing seed exchanges. So in Southborough, they collected seed from the Penstemon Hercetus, the showy goldenrod, some of the harder ones to get. And they did a winter sow event and they got the seedlings and they put them in, and a lot of those plants are doing really well. So a lot of what I'm doing is to provide seeds and cuttings, if it's a willow, to give to people. So if you have problems finding a plant species that you want, just contact me and I'll point you in the right direction. I hope more people, more nurseries get some of these plants. They are difficult, but I've managed to get pretty much everything on my list. So.
And there are some native plant nurseries in New Hampshire as well that are working really hard to produce, oftentimes from seed.
Right. So I'm not as familiar with those. So it's not like I'm trying to diss them. I'm just unaware of them. So if you wanted to send me good sources, I'm happy in my talks to, you know, if somebody says that they're from New Hampshire or you know, it's a short drive, I'm happy to promote other nurseries as well.
That'd be great. Are there resources that share the pollination system, both the plant and the pollinator? Beyond, it sounds like your website has some. Are there other resources?
Not really, no. You know, unfortunately, the native plants have been neglected or separated. So the typical way of doing things is to, you know, you put out kill and pin, looking at the bee abundance and not thinking about the connection with the native plant, and certainly not looking at native plant pollination and native plants that are in trouble, and whether it's because the pollinators are declining as well. So again, I'm hoping that what I'm doing will make more people aware of this from the research side and appreciate and fill in some of the gaps that we have.
So Rob, can you determine a pollination system in your backyard, in your garden, in your pollinator meadow through observation?
Excellent
You're
question.
doing
Yes,
close observation?
you certainly can. You can look to see if the animal is making contact with the male and female reproductive. So you need to be familiar with your flower morphology. Varies with species, but you can identify the male and the female part, and you want to see is there contact being made with both. And the workshops that I give, I show people. So last weekend, there's Monarch officiosa is in bloom, and I was showing people, look at this big long-tongued bumblebee is touching, and these smaller bees and some of the butterflies aren't touching. So here the butterflies would be flower visitors, but these bumblebees are acting as pollinators. And so, you know, a lot of this is attention to detail, right? Knowing the difference between the species, knowing the plants that you have and how, you know, are they making contact? So close-up observations can help.
Are your workshops, your in-person workshops where you're doing this on your website?
I try to put my talks and workshops on my website, but I'm not great at that. I sort of, it goes in bursts. In terms of holding workshops, you know, just email me and I could hold you know, have a workshop where we can go over some of these things. My focus now is assessment. So how do you assess the quality of the species that you have and show people, you know, that putting in Black-Eyed Susan is not helping much. Even though people love it and it looks great, it's not fun. Like if you were to look at Monarda versus Black-Eyed Susan, which, you know, at this time of the year at the workshops that I did, it's very clear which one's helping and which one really isn't. So I'm happy to hold these workshops if you were interested in organizing one. I think this year, for next year, certainly is doable. I'm trying to think of the next one I have coming up. It's in Rhode Island, so it's a bit of a drive. I have one on Saturday in Rhode Island.
We'll definitely keep you in mind for next year.
Yeah.
That would be great. So if somebody is using your Beecology web app,
Yeah.
and they send information in, they take a video and you get it, what kind of feedback will they get from it?
So I go in and check the species ID and the sex, you know, and the flower, and we'll correct the plant species if needed. If the plant species is marked as unknown and it might not be on the list, I can add it. But we're just, you know, I've got a meeting tomorrow about deploying this comment function where you can add a comment, and then that I will see that comment and I can reply to your comment. So when you retrieve your logs, you'll be able to see the comment that I've made and any correction. So that function's coming this week. Before that functionality gets incorporated into the web app, just send me an email. Say, I just sent in something, could you help me? And a lot of people do it that way. And if you don't want to use the app, right, the video, that's your data. Just send it to me or create a Google Drive and put them in. Any way you want to get that to me is fine. The web app just makes it easier in terms of what I need to go through and file size, with problems with file size and emails and things like that. But
is there also a good website differentiating bee species of all kinds?
Yeah, I mean, there are some, there's Bees of Eastern North America, there's a PDF, USDA put that out. Each state, I think New Hampshire, there's definitely a good source for being able to tell bumblebees and generally other bee species. A lot of you can generally, for the non-bombus, it gets tricky because a lot of them are small and hard to separate based on species. The butterflies, there's the Mass Butterfly website that is great for IDing the butterflies.
So I'm just curious, does it help you at all if you're doing some methodology when you're taking these videos, meaning the same time of the day, or varying times of the day, or on some kind of an interval? I'm
Right.
just curious.
Excellent question. So yes, we want different times of the day in case there are temporal patterns. Typically what happens is that the nectar sources build up overnight. I mean, there is some definitely moth pollination, which we're looking at, moth visitors, but Most of the nectar's in the morning, and so you'll see a lot in the morning, and then as the day goes on, things can change. So you want to do the same time, but it's also good to check other times just to make sure you're seeing the same thing. But I have a bunch of ecologists that are doing intensive surveys of their property, and they're telling me what else is in bloom. So it's not just what's important isn't just what they're visiting, but it's what they're not visiting that is equally important to me. So if you know there are 6 things in bloom and they're only on one thing, that tells me preference. If there's only one thing in bloom, it may be because it's the only thing in bloom, or it may be because there's a preference. So giving me that type of information certainly helps me to determine whether it's a preference, and how many bees you're seeing at a site, whether they're different individuals is important, because there are one-offs. As I mentioned, that you might see a bee on a species that isn't on my list, but that's because they're not going to starve. As I said, they'll eat fast food, they'll try things rather than starve, but that doesn't mean that they prefer that source. And the preference suggests that they do better for whatever reason, and we're trying to figure out those reasons, but we try to, as we try to determine the preferences, that's the way that I go about it. And so yes, you could help that way, being more rigorous with the surveys.
Okay, and it sounds like you like a lot of detail if possible.
Of course, the detail is where we get like that, those are the data. The more detail, the better picture I get, because when I get the same set of conditions in Western Massachusetts and see the same thing that you're seeing in New Hampshire, then those patterns, if they hold throughout the range of the species, that's important, because then I can tell people in Canada to do what I'm doing in Massachusetts, and they'll be helping as well.
Great. Here's one more specific question. There were several pictures of scarlet bee balm in your presentation, but it's not on your list, correct?
Right. So there's a Monarch officiosa rubra that is, I believe that it is a subspecies. It's not a cultivar, and that should be so. The tongue length, I don't have enough. So you can see I'm fumbling. So the didyma. There are mostly cultivars of didyma that are out there, but the tongue length is it's a bit too long for the tube length, is a bit too long for Fervidus anaphanes. The rubra it looks red, it looks like a didyma, but it's got a shorter tongue and they're all over it. So the reason that the didyma is not there is because I haven't seen the numbers on the didyma, certainly compared to the fistulosa. So that's why it's not there. Okay.
So one more question. You don't mention any shrubs. Are they also important?
I mentioned tons of shrubs. So northern bush honeysuckle is important. All of the so meadowsweet is shrub. The
willows,
native roses, the willows, you can get shrub willows. The meadow willow and dwarf prairie willow are great additions. They like drier soils. So go down the list. There are definitely shrubs on the list. In fact, in the spring, shrubs are what's carrying most species. Early spring, it's trees and shrubs.
Great, thanks, Rob. I should say there are many positive comments in the chat. Thank you for the great work you're doing. Thanks so much for the interesting presentation. Many of those types of comments, and I'll just share out with the attendees that there are a number of different nurseries that were placed in the chat as well. So if you want to take a look.
Great, thank you.
And thanks, Rob. This was great. And we'll definitely look for you next year with the potential for doing something in the field. And I will throw it back to Mark just to wrap things up. Okay. And again, a big thank you.
Thank you. Thank you, everyone, for attending.
Yeah, that was great. So informative. And thank you all for sticking with us and asking such really interesting questions. I shared a link to the webinar survey in the chat, and I'll also send out a follow-up email with some of the links that were shared in the chat as well. The next talk will take place on August 24th by our humanities expert for the project, Dr. Maria Sanders. So please visit our website to register for that talk, as well as the rest of the upcoming talks. And as a reminder, we'll be hosting these webinars through February, I think. So thank you again, Rob, for presenting this evening, Diane for organizing this series, and for you all for tuning in to learn alongside us. We look forward to seeing you again at a webinar in the near future. And as always, for ways to be get involved as a member, volunteer, or donor, please visit us at nhaudubon.org.
All right.